In the last session on biosensors, we discussed various types of sensors, and the previous one we mentioned was about the biosensors that is used for the detection of heavy metals. Now, we will discuss the biosensors that is used for detection of nitrates and nitrates. Here, at the bottom portion, there is an equation of the conversion of the nitrate and nitrate is given. First of all, there is a nitrate that is reduced by nitrate reductase to nitrate. So the NO3 minus, that is a nitrate, is reduced by nitrate reductase to nitrate, that is a nitrate, that is a NO2 minus. Then this nitrate is actually reduced by nitrate reductase to ammonia. So this is the biochemical reaction that is occurring during the reduction of nitrate and nitrate. So in the case of biosensors that is used for detection of this nitrate and nitrate, we will start with the first one in this case, where we have nitrite as the analyte. You will use glass beads which is immobilized with a nitrite reductase. So the nitrate will be converted by the nitrate reductase, that is the second step, to ammonia. But when your pollutant or the analyte that needs to be detected is nitrate, that is the first one, then it needs initially to be converted to nitrate, then final conversion to ammonia from nitrate needs to be done. So in this case, the glass beads were immobilized with the nitrate and nitrate reductase, that is both enzymes were immobilized in the system. So the entire pathway of a conversion of nitrate to nitrate and nitrate to ammonia can be completed within the same immobilized system. This is how nitrate and nitrate reduction mechanisms works. So a biosensor, usually if you want to detect nitrate or nitrate, the presence of nitrate or nitrate, you need to immobilize the concerned enzyme, that is a nitrate and nitrate reductase, into glass beads and use it for the conversion of that nitrate or nitrate to ammonia. So this is the process that is occurring. We have basically two analytes, that is a nitrogen compounds, that is nitrate and nitrate. So the biocatalyzer that is generally used is from C-based biocatalyzer, and which is actually used in an amperometric biosensor, which is nowadays widely in use. Then Viologen mediator based nitrate sensor is there, in which the electrochemical detector, electrochemical mechanism based detector is actually used. Usually, the toxicity of the nitrate is based on the formation of a methemoglobin and of the carcinogenic N-nitroso compounds, which is produced by the reaction between the nitrate and the secondary or tertiary amines or amides which is present in the system. So the interaction between the nitroso compounds and the nitroso compounds is actually the carcinogenic nitroso compounds is produced by the interaction between the nitrate and the amines or amides which is present in the system. So we have the formation of a methemoglobin, which is one of the toxic compound, and also the carcinogenic compounds like N-nitroso compounds, which is by the interaction between nitrate and amines and amides which is present in the system. So this is why we need to detect the presence of the nitrate and remove it subsequently. Whereas the nitrate or NO3 minus, that is as such it is not toxic, but as you know in the previous reaction, the reduction of the nitrate will produce a nitrate which is having toxicity, as we mentioned in the previous paragraph. So generally, if you are going for detection of the nitrate or nitrate, both is relevant because the nitrate, as such, if it is not toxic, then also there's a possibility of this conversion of nitrate to nitrate and cause such a toxic effects and carcinogenic effects in the system. Then detection of nitrite and nitrate can be performed using an electrochemical assay with nitrate and nitrate reductase combined with the reduced form of a methyl vialogen as an electron donor. So in the previous diagram that we see here, we use the glass beads which is immobilized with the nitrate and nitrate reductase, and then we will use an electron donor, something like methyl vialogen, because an acting as an electron donor for the detection system. Column with the immobilized nitrite reductase serves for nitrate assay, and a second column with the immobilized nitrate and nitrite reductase indicates the amount of nitrate plus nitrate. So for the actual detection, you need two different columns. One with an immobilized nitrate reductase, and the other column, or the second column, with a combination of nitrate and nitrate reductase. Why it is so? Because if you want to calculate the quantity of the nitrate in the system, you need to subtract the actual quantity of the nitrite present in the system, because nitrite is an intermediate in the conversion of nitrate. So already existing nitrate, if any, in the system needs to be initially detected, and then it should be subtracted from the nitrate, because the conversion of nitrate will produce nitrate also. So that two nitrates needs to be distinguished between when we are calculating the quantity of nitrate, because the end product is same ammonia. So the ammonia produced from nitrate needs to be subtracted from the ammonia that is produced from nitrate. So that's the concept. So both the systems, or both the columns which is immobilized with the nitrate reductase and another column with the nitrate and nitrate reductase needs to be used, and simultaneously the same sample will be detected with the two enzymes or enzyme immobilizers. And finally, if you want to assay the nitrate, then directly you can take the column nitrate reductase. And if you want to assay the quantity of the nitrate that is present, then you will use both the terms, and then finally you will reduce or subtract the actual quantity of the initial nitrate which is present in the system, which is detected through the nitrate reduction column. So that's how the quantity of the nitrate is actually detected using these biosensors. Then there is another compound that is a cyanide, which is also a toxic compound, which needs to be detected in the presence of which needs to be detected in the environment. And here we have immobilized rhodanase or injectase, which are the enzymes, and that can be applied in the enzyme thermistor for cyanide detection. So in the immobilized system, the enzymes that is used is a rhodanase and injectase. And usually it produces a heat signal. The heat signal generated by the conversion of the cyanide, and it catalyzed cyanide catalyzed by the immobilized enzyme, leads to a relationship of about 20 to 600 picomolar cyanide with the injectase, and about 20 to 1000 picomolar with the rhodanase. And the range of 20 to 6000 or 600 or 20 to 1000 picomolar, and that is the actual detection range of the system. If you are using injectase as the enzyme for cyanide detection, you can detect about 20 to 600 picomolars of the cyanide, or as low as 20 picomolar of the cyanide molecules present in the sample can be analyzed using injectase system. And the other one, with the rhodanase enzyme, that is also is having the same sensitivity of about 20 picomolar, and you can go up to 1000 picomolar detection range. And in that range, there is a linear relationship, because if the just like the sugar estimation graph or the standard graph that you're plotting, it should be linear. So the concentration or the change or the response should be linear. Then only you can predict it. But when the linearity loses, then your model will not be valid anymore. So the prediction or the calculation will not be working in a range above or below that sensitivity range. That's why there is a linear relationship. We are saying that there is a linear relationship between the interaction of this system. Anyway, as I mentioned, the two enzymes for the detection of cyanide is rhodanase and injectase, and both having about a detection limit of about, within the range of picomolars, 20 to 600 picomolars, which is very good when we consider the quantity of the cyanide, because cyanide toxicity, if it is present in even picomolar, then that needs to be relevant. So it needs to be detected at a very low concentrations when you are comparing with some other analytes, may be detected in milligram levels or millimolar levels. Now, besides the thermistors, electrochemical sensors are also in the laboratory use for cyanide detection, and which is also in combination together with, it is in combination with the rhodanase as the biological component. So rhodanase enzyme is actually acting as a biological component for the electrochemical sensors, which is actually developed in these days. Now, there is another analyte, that is a sulfite and sulfide. A sulfite ion sensor has been built using immobilized organelles of the organelles and an oxygen electrode. In this device, a linear relationship between the steady state current and the cell concentration, the linearity of the standard graph that you are mentioning, was obtained up to 340 picomolar. So that is the detection limit. And if you are going even up to a low concentration of 340 picomolar, then also you will get a linear relationship because the prediction will be correct. And the time necessary for detection is about 10 minutes, which is also good when you are comparing the analysis of some environmental samples. Usually, the detection time or the time required for getting the results is a very important parameter in the case of such a sensors. So, if you are getting the results within 10 minutes, then that is very good. Then, a photomicrobial electrode for selective determination of sulfide has been tested by some scientists, Matt Suna et al. In 1984. So there's a photomicrobial electrode for detection of sulfide. They use the photosynthetic bacterium Chromatium species on a hydrogen sensitive electrode. So one of the examples of a photomicrobial electrode, which is used for a sulfide, is the use of a Chromatium bacterium on a hydrogen sensitive electrode. And this leads to a sensor with a response time between 5 and 10 minutes. So even in the previous case, it's about response time is about 10 minutes. And that detection or the time required for detection is actually again down by these photomicrobial sensor to about 5 minutes, or you can detect within the time of 5 to 10 minutes. And linear range is about is in the range of a millimolar, and that the previous example, your detection is about 340 picomolar, which is very low concentration. But using my photomicrobial system, it's about 0.5 millimolar. So you can detect in a manner, but detection limit or the analyte concentration that should be about 0.5 millimolar. So that is an issue with these photomicrobial electrode sensor. So compared to the previous one, the detection limit is it is in millimolar range, not in picomolar range. Then sulfide could be monitored with an oxygen electrode after fixing of a Thiobacillus thioxidans cells by a gas permeable membrane into onto a probe. So there is another example is a use of Thiobacillus thioxidans, which is actually fixed to a gas permeable, and that system can be used for detection of sulfide. So that is another is about monitoring of organic substances. The main organic substance that we need to detect is a phenolic compounds. The environmental pollution by substances is a widespread. It is widespread, and because of their intensive use in the industrial chemicals, for example, for the production of polymeric resins or pesticides. They are and the phenolic substances were actually used for the production of such a pesticides and polymeric resins. So it is a serious environmental issue when you are considering the environmental pollution. Many biological compounds have been tested for the construction of phenol biosensors. And microbial sensors have been developed with the trichosporin cutanium. The organism that is used is trichosporin cutanium, and with a linear range of about 0 to 15 milligram per liter of phenol. So that's a detection limit or detection range of a phenol detection using sporin cutanium. And with two different strains of Pseudomonas putida, with a linear calibration curve of 2 to 11.7 picomolar phenol and 17 to 34 picomolar paranitrophenol. So different phenolic compounds can be detected with different organisms in various ranges, ranges from milligram to picomolar range. The combination of an Acetobacter species with an oxygen electrode was used for the determination of a phenol and hydroquinones, whereby linearity was about 2 to 16 milligram per liter range. So detection limit or the range of accurate detection will be 2 to 60 milligram per liter. So there are various systems for phenolic compound detection. Trichosporin cutanium based system is there. Pseudomonas putida, two systems for phenol and paranitrophenol. Both systems are there. Then Acetobacter based system for detection of phenol and hydroquinones are there. And these are all good systems for detection of different phenolic compounds. Then, am I audible? Hello?
Yes, sir.
Yes, sir.
Yes, yes.
Yes, sir.
Then a biosensor for 2-ethoxyphenol constructed with rhodococcus, another organism, and an oxygen electrode also gave a linear range of detection between 0.05 and 0.4 millimolar range. So there is another system for a 2-ethoxyphenol, which is another phenolic compound, and the organism that is used is a rhodococcus. So many enzymes which catalyze the oxidation of a phenolic substances exist in nature. For example, in organisms that phenolic compounds, such as or secondary metabolites, such as alkaloids or tannins, these therefore can catalyze in the enzymatic phenol sensors. So various natural enzymes which is actually used for oxidation or oxidation of phenolic compounds, microbial derived or different environmental derived, different enzymes which actually oxidize the phenolic substances were existing in nature itself. And these naturally existing enzymes can be usually that is used for the production of lignins and secondary metabolites and different alkaloids, or the phenolic compounds will be converted into compounds. And that enzymes which convert the phenolic compound into different alkaloids and secondary, that enzymes can be immobilized and can be used for developing phenol sensors. Tyrosine, for example, it catalyzes the orthohydroxylation of phenol to produce catechol. So the phenol is orthohydroxylated using the enzyme tyrosinase, and that produces catechol. Let's say catechol is an intermediate in the phenol degradation pathway. And the dehydrogenation of catechol to orthoquinone, being converted to dehydrogenated to orthoquinone. So that's a intermediate compounds which is produced in the conversion of phenol to similar forms. In addition to phenol and catechol, it can also oxidate para-substrate phenols, tyrosinase enzyme. The tyrosinase has been used in combination with an oxygen electrode. However, lower detection limits can be achieved by amperometric detection of the catalytically generated quinone products. So there are two systems. One, it can be detected using usually the thyroxinase we combine an oxygen electrode. Higher detection limits can be achieved. Amperometric detection method, which is based on quinone products, which is a final product, not goal based or initial stage detection, but the final or late stage product like it can be using amperometric mechanisms, which will give you results. So at various stages, all degradation be detected. Even in the initial stage of the conversion of the catechol or at the later stage of a production of quinone substances, both the systems can be used. Oxygen electrode based systems or amperometric systems can be used, and the system will actually give you better detection limit. So here it summarizes different types of analytes like biosensors, phenol, different compounds like catechol, then cresol, chlorocresol, then M-cresol or catechol, then so different compounds are listed. And these different analytes can be detected using the catalyzer. Lactate or tyrosinase is one of the enzyme which is used in amperometric system. Then DNA-based detection system for M-cresol or catechol used in amperometric. All these are amperometric systems. Then mushroom tissue, which are produced tyrosinase that can be used for the detection of phenol. Then oxidase that also be used. So these different detection systems we all analyze biocat combinations and the corresponding detection system, which is also mentioned there. So here, as I mentioned, you can detect the conversion of phenol to one or the initial stage conversion that can be detected using amperometric system, or you can use a potentiometric detection system, which a latest conversion of the quinone to similar forms. This is glucose dehydrogenase enzyme. This is the lactase enzyme, actually converts the phenols to quinones. Then the quinone compounds is actually broken down into gluconolactone by the glucose dehydrogenase enzyme, and this the detection product will be gluconic acid, and it can be detected using the potentiometric detection system. And we can use the oxygen consumption using an oxygen, which is used in amperometric detection system, and the phenol to quinone, phenol quinone, that conversion. During the conversion, the oxygen electrode will detect the conversion. So that's based on amperometric detection system. And at the later stage, the degradation of quinone compounds to gluconic acid that can be detected using the enzyme immobilized glucose dehydrogenase enzyme immobilized system, which is actually based on a potentiometric detection system. So some of the phenolic compound detection systems. Now, the next one is about the pesticides. For the detection of detection and quantification, or for the identification and quantification of pesticides, enzyme inhibition can be used. So the theory or the principle behind the detection of these pesticides is the enzyme inhibition, and that can be used for analytical methods with high and selective, selective sensitivity. So these are chlorine esterases. which may be acylchlorinesterases or butyrylesterases. This cholinesterases, that enzymes, acetylcholinesterase or butyrylcholinesterase are the examples, play an important role in the detection of pesticides. Organophos and the carbamate insecticides act as inhibitors of chlorinesterases. Usually, chlorinesterases are the enzymes which is actually used in this system. That or the analyte that needs to be detected, which are phosphorus compounds and carbamate insecticides, they inhibit the esterase activity of the enzyme, thus leading to a decreased pH and after the addition of the corresponding choline ester. So, in a system in which pesticide present or the analyte, if the analyte or the sample actually it contains the analyte or the pesticide, then if you add the cholinesterase, acetylcholinesterase or butyrylcholinesterase into the system, it will inhibit the pesticide present will inhibit pesticides were organophosphorus compounds or carbamate insecticides. They will inhibit the activity of the cholinesterase that is added, resulting in a decrease in the pH change. Usually, the activity of the cholinesterase will be there, should be there, and that activity is actually prevented by the insecticide or pesticide that is present in the sample. The inhibition by various pesticides differs strongly and is in the presence of a mixture of different pesticides. It is not proportional to the sum of the pesticides. So, if there is only a single pesticide sample or a test pesticide that is present in the system, it will give you a linear result. But normal environmental samples may contain a mixture of the pesticides. So, it won't give you a correct response. You can't predict the actual output based on the inhibition of the enzyme, because if it's a mixture of 3 different pesticides which is present in the sample, and the pesticide A may contribute to some amount of inhibition, B and C will have its own range of contribution to the overall inhibition of the enzyme. So you can't differentiate in this. So in a system in which of the insecticides or pesticides is present, you can detect the overall effect of the insecticides. That is the organophosphorus or carbamate insecticides were present or not. You can find out the overall effect, but individual compound detection is not possible with such a enzyme inhibition assays, because the group of compounds simultaneously inhibits and each will have its own inhibition range. So in the mixture, you can't actually quantitate which compound or which organophosphorus or carbamate insecticide is present in the system if it is a mix of insecticides that is present. Cholinesterase biosensors can be used for a screening test to establish the presence of cholinesterase inhibitors and indicate a more detailed investigation. So the thing that we can conclude is that there are different cholinesterase inhibitors. So the sample contains cholinesterase inhibitor like organophosphorus or carbamate insecticides. So the sample needs to be investigated thoroughly or investigated in detail. So that is the only outcome that we can achieve. So acetylcholinesterase has also been used in organic solvents for the detection of pesticides. So these are the table actually shows the analyte and biocatalyzer combinations and their corresponding detection systems. Paraxone, parathione, carbaryl. So these are different analytes or insecticides or sites, and or corresponding enzyme combination is cancer. The detection system, maybe that is optical fluorescence, electrochemical or amperometric systems were used in combination with these analyte biocatalyzer combinations. Then the next next table actually it shows biosensors used in the detection of herbicides. There are Phenoxyacetic acid, then diuron paraquat. These are two herbicides that is used, and the type of interaction is actually the first one. In the case of the dichlorophenoxyatic acetic, there is a immunoanalysis based interaction is there, and the second one biocatalytic interaction is there. The biocatalyzer is acetylcholinesterase is the first enzyme. The second one is a cyanobacterial enzymes who are used in the second detection system. So the combination of the analyte and their interaction or the biological component is actually mentioned there, and the detection system in which it is employed is also marked in the table. So this is about the biosensors for the detection of a environment sample.